from, you did?
No.
In your little?
I got that myself, and I got that half for me, and then the other half for her.
That is so cute. Did you get it from like that convenience store?
It's like Etsy or something.
I don't know, they were advertising this at Six Flags one time, and I fell in love with them. They got their car, and oh yeah, I love chocolate. Everything.
Yeah, it was like Etsy. Because you could get like a really funny word. You would be like,
So many things like that.
it's kind of weird to have big on my finger though.
I
Because
don't
you're not like sorority, just outside. The good news is you're probably wearing like 3 other things that say Alpha B on them at any given time, so.
Just study.
Yeah, exactly.
The bronzes.
That was a good time to see it.
You just are over there, not even.
She's ripping lines.
I'm sorry, I'm wearing the same thing I wore yesterday. I didn't see it.
I'm wearing the same thing I wore yesterday.
You're like, oh sweet, well this is put this on, I'll just keep this on.
I didn't really want to change this morning.
Well, I just woke up at the open house and thought I was going to yoga, so I kept like clothes on when I could do yoga. Then I got
You would not believe the
so-
pile of hair.
No, I'm just not wearing.
It was a tremendous amount of hair.
I'm just gonna wear this till my show.
You need to like walk around. The trash can. I hope so. Okay. This is where we were last time. There's a tremendous amount of information in that image. So I'm going to walk. Last time we started at protein, we worked our way up. Now I want to give you this. Are we on or are we rolling? I want to give you this and work our way down. So let's refer back to central dogma of biology. So let me start by asking you some questions, and you think you're going to know the answer. When we walk our way through it, maybe it gets you to think about things a little differently. Okay? Where is the transcription start site? +1. Where's the translation start site? Is it on here? Yes, it is. Where the shift from white to black goes. Right? So, who wants to come up? Who's bold and wants to come up and draw the pre-RNA? Someone come up and draw the pre-RNA for me. Appreciate that.
We're using these.
Yep. Thanks, Michael. Appreciate that.
Is that the stop?
This is the terminator.
Yeah. That's the 20.
Okay, great. I agree with that. I'm not sure I put this on. Think about that. Yeah, I guess the terminator would be on there, wouldn't it? Okay. So what we've got is the transcription start site +1, and we go all the way down to the terminator. It's actually supposed to be longer, but it's not really perfectly scaled up under that. Can someone draw someone different come up and draw the mRNA? So that's the pre-RNA. Can someone draw the mRNA? Jacob, way in the back, you want to do this for me? Okay, that's what you get for sitting back. What's your favorite color?
Let's go with green.
Green. pretty good. I'm going to draw one extra thing on here, which we haven't really talked about yet. That's fine. I'm going to put a note on here, cap, and note the poly A tail. And I'm going to say probably most of this gets cut off. And put the poly A tail in. All right. Who's going to draw the protein? Come on. Fill up your head.
Which one?
That's pretty easy, isn't it?
I guess we don't include the, like, that tidbit of.
That's it. It's the protein. Right? So that codes for the amino acids. This would actually be 1/3 the size, because right, 3 nucleotides codes for 1 amino acid. We draw it like that. Okay, great. So what we've got is, I'll put the AUG up here. And this is going to be the termination. It's either TGA, TAG, TAA, correct? Okay, so what's this thing?
Promoter.
Promoter. It's not intron 1, exon 1? Promoter, great. Great, okay. So hopefully in practicing this has helped you along on this step. So up to now we've talked about splicing. We've talked about this dystrophin gene, 2.5 million base pairs. Gets cut down to just 14,000. Talked about after splicing, what happens is we have a whole bunch of proteins that bind at the intron-exon boundaries. They help recognize the splice sites, those consensus sequences. In the messenger RNA, those proteins actually remain bound. Some of them. Like that. Okay? We can talk about that a little bit later. I think I talked about it later in the class. Okay, other types of modifications. There are 3 things that happen in processing. You should know this. Splicing, capping, and polyadenylation. In lab, we already talked about polyadenylation. That's great. Capping. This is when I told you that we add nucleotides onto the 3' end, right? So we have our sugar. We have our base poking off like this. So we got 3 phosphates poking off like this. This is the 1, 2, 3 carbon. So this is my 3' hydroxyl. And the next nucleotide is going to come in. The base pokes off there. This oxygen is going to come across here. We make this connection. This pyrophosphate goes away. And we have our 3 to 5 connection here. This, the phosphodiester bond right there. Of course, we can add the next one on here. So that's why we write our sequence from the 5' to the 3'. It's the order that it's created. This is the 5' end. This is the only time, to the best of my knowledge, this is the only time that anything is ever added onto the 5' end of a nucleic acid. What we get, I'm going to draw it the same way we did before. We actually have a guanine. So it's the same nucleotide that you would put in DNA here. Like this. And this guanine is put on backwards. And the specific details, how many phosphates do we end up in there? 3. So one of these guys pops off, 2 of these guys pop off, and we connect this way. Normally what we would think, the normal connection would be guanine off here and our phosphate's up like this, right? Normally, we would think we'd get this connection right here, right? But we're connecting this phosphate over into this phosphate. So this guanine is on backwards. The effect of this is I now have a 3' end here and a 3' end here. I don't have a 5' end anymore. Does that make sense? We still call this the 5' end. But there's no exposed 5' because this is the 5 right here. Just go red. This is the 5 carbon right here, right? And this is the 5 carbon right here. So what happens is a guanine gets popped on the 5' end backwards. And for reasons I don't know, well, I can speculate. The guanine then gets a methyl group put on it. So the 5' end of a messenger RNA has what we call a 7-methyl guanine cap. In the guanine, the 7 carbon in the guanine gets a methyl group on it. If you said it's a guanine put on backwards and left it at that, I guess I'm okay with that. Go to grad school and talk about the specifics of where the methyl group is. But I'm going to say 7-methyl guanine cap so many times that it should just flow out of you. The Zen of the 7-methyl guanine cap. It's a part of you now. Okay? Why bother putting a cap on the 5' end? Does anyone know? Has anyone looked at the PowerPoint file and read why? Yes? One of you?
Unstable.
Stable?
Yeah, it's unstable without it.
It's unstable without. Why is it unstable without?
Because it has the phosphates hanging out.
Okay, yeah, so I mean, we could degrade the phosphates off, who cares? So if we have our 5' end, then we degrade the phosphates off, so what? Do we need to have those phosphates on there? Yeah, you're saying yes? Do you know of a reason? Because I don't know of a reason. We need the phosphates on, so we can put the 7-methyl guanine cap on backwards, right? It is for RNA stability. Why would having this on make it stable? You know, if we took those off, who cares? To the best of my knowledge, it doesn't do anything.
Couldn't it bond with like free nucleotides?
Could it bond with free nucleotides and maybe you add nucleotides on the end? I hadn't thought of that, maybe. Turns out, it's part of your immune system. What? So human cells have the ability, because we've evolved a machine that puts a 7-methyl guanine cap on here. A virus doesn't code for that machine.
Ooh.
So a viral RNA has an exposed 5' end. So what we have evolved to protect ourselves from viruses is we invented a machine that would stick a cap on there, and then we invented a machine that destroys RNA that doesn't have caps on it. How cool is that? So what we do is we make an RNA, and on the RNAs we make, we put a cap on it. Then any RNA that doesn't have a cap, we just go and destroy those RNAs. Voila, protection against viruses. Until viruses evolve ways of stealing our machine and putting caps on their RNAs. But most viruses don't do that yet. Okay, cool. Seven-methyl guanine cap. And then in lab, we talked about this AAUAAA, the sequence, which basically there's a protein that binds to that. What we'll learn about today? Yes, today. And when is it? Thursday? We have lab. Today, Thursday in lab. We'll learn that there's another protein on the other side of the cut site that binds. And the two proteins interact with each other, and they cut it, and then there's a poly-A polymerase that adds a whole bunch of A's on it. Why? I want you to think about what we did in lab. Why did I ask you to look for this consensus sequence last week, and I didn't ask you to look for the consensus sequence here?
Because it was already cut off. There was a poly tail on the right.
Yeah, I had just defined it for you. We make our RNA, and we have the A, U, A, and then we have a sequence which is full of G's and U's. In this area, there's a U. I said there's a protein that recognizes this consensus sequence. There's a protein that recognizes this consensus sequence, and they say, hey, we're going to cut right here and destroy this and put a bunch of poly-A's on here. So last week, we're looking at a cDNA sequence. You were looking at the processed thing. You're not going to see the GU-rich sequence. It was cut up. On here, someone want to come up and draw for me where that is? Anyone? Want to take a chance? You guys don't like this getting singled out in business, do you? Come on, someone, please.
Draw where the poly A tail is.
No, draw where the GU-rich sequence is. Well, draw the poly-A consensus sequence, the AAU, AAA. Where's that? And then draw the GU-rich sequence. Where that must be. You want to do it? You will try. We will not heckle you to that. We can heckle.
Is that right here? Is it AA?
Yeah, the AAU, AAA, somewhere in there, yeah.
And the
GU-rich.
G.
That's it. Is that what you guys were, you were all thinking that, weren't you? From the nods, I can just tell. And all the smiles.
Why is it a poly A tail? Why not like T or something other?
That's a great question. The question is, why is it a sequence of A's? Why isn't it a sequence of T's? Why cells chose adenine? I don't know. It might be that ATP is more prominent, so A was easier to get a hold of. That's maybe why it evolved to be that one. From a practical standpoint, the reason it is A is once it became A, we evolved our cells to use A's. And if you put something else on there, it wouldn't work. Why it originally became A's instead of T's, I don't know. Can't give you an answer. Yeah?
Why isn't it in the part that's actually translated? Because right now it's in the part that's not being translated at all, because right now we have it put in a part that's not actually going to end up in your mRNA. Like what?
Oh, yeah. You're right. You're right, and we were wrong. Thank you. Yeah, it has to be transcribed. So what Sammy said is, how could it possibly be here that it would be used to cut the RNA? It suggests it's in the RNA. But if this is the terminator, then you would never get in the RNA. So the GU-rich has to be here. Right? Now it's in the pre-RNA. And the AAUAA is somewhere in here. So that it will go and it will cut in between here. And the way I've got this, I guess we'll cut that off like that, right? So this stuff, we're not up here in there. Yeah? Thank you for that correction. You get a gold star today. I promise I will not call on you for the rest of the week. That was awesome. If I had written an exam, I would have written the exam question incorrectly. Good for you. Okay. Any other questions on the poly-A tail? So what happens is the cut happens and there's a polymerase there, And it adds a couple adenines, a couple goes along real slow apparently. And then once it kind of gets in its groove, it's got a couple beers in it, it's on the dance floor already, and off it goes and it adds like 2, maybe 300, 350 nucleotides on all adenines. So it's a very, very long stretch of adenines on here. And we generally say about 250 adenines onto the 3' end. Okay,
So is the poly A tail like right after the terminator then?
it's before the terminator. The terminator gets cut off and thrown away.
But you said there's A's that connect to it. So like once we cut off.
No, so what happens is, yeah, so the terminator is over here like this. When we put the poly A tail on here, this thing gets just degraded. Right? So we put the cap on here. Does this make sense?
Well, I thought you said like we take the GU rich portion out and then it connects to the rest of the A, which I was assuming would be after.
So there's a protein that recognizes this consensus sequence. Thank you for asking. There's a protein that recognizes the GU rich sequence. And it cuts in between them, breaks the covalent bond, so this is now its own RNA with the GU rich in it. And then the polymerase is associated here and has a bunch of adenines on here. What's this end? Is that 5 or 3?
That would be 5.
The 5' end. Does that have a 7-methylguanine cap on it? No. So it must be a viral RNA we should destroy.
I'm saying like all those extra adenines, do they just come floating in there? Or
No, there's a polymerase on here.
Okay.
It says, here's my 3' end. I need an adenine. Give me an ATP. Bam, got it. It just totally adds them all on.
Okay.
And adenine is just all over the cell. So that's probably why it was chosen as the nucleotide. Okay. So the cell has machines that degrade unprotected RNA ends. So we've got our 7-methylguanine cap, and this end is protected, but this is a 3' end, isn't it? We got a 3' hydroxyl over here. Right? This is going to suffer the same fate. The machine that destroys RNA is going to come and say, oh, 3' end, I'll just eat my way this way. Right? So we need to protect that end. And the poly A tail isn't the protective cap on there. It turns out there is a protein. I'm going to give you the name of the protein. Anyone tell me the words that protein represents? Go for it, either one of you guys.
Poly A binding protein?
The poly A binding protein. What it does is it binds to the poly A tail. I want you to think of this. Have you ever been walking in the woods? Like I did one time stupidly with no shorts on, walking through the brush, and when I got out, the hair in my leg was covered in burrs. Can you imagine how the hair in my leg was all knotted around on these burrs? So the poly A binding protein binds to adenine very tightly, and it just kind of makes this big mess of a snot of the poly A tail. So that the poly A tail actually kind of, I'm going to do it in a different color. The poly A tail kind of wraps all around because the poly A binding protein, there's a whole bunch of them, maybe 100 of these guys, all bound up. And in that mess of stuff, where the hell is the 3' end? It's kind of hard to find the 3' end for the machine to come in and destroy it and tear it away and degrade the RNA. And that is how the 3' end gets protected. Questions on this? Okay, good. Okay, good. That was actually supposed to be wrapping up yesterday. Now, we're on to the translation. I did not have a chance to review this. I was grading this morning. So, I'm going to kind of wing it here. So we actually learned that there's 3 different kinds of RNAs in the cell. There's transfer RNA, there's messenger RNA, and there's ribosomal RNA. Ribosomal RNA is the RNA that's part of the ribosome. The ribosome is the machine that translates. Transfer RNA, I call it the T for taxi RNA. tRNA is transfer RNA, but I use T for taxi RNA. It goes to the amino acid bank, takes amino acids out of the bank, and takes them to the ribosome to build proteins. Okay? So there are actually 3 different types of genes. And when we first discovered genes, or were conceptually thinking of genes, we said there must be a code in the DNA, and that code is translated. So every gene must be translated into a protein. So when people discovered transfer RNAs, you make an RNA, and then the RNA folds up in a 3-dimensional shape, and that's it. That's the machine that does the work. It blew everybody's mind, like, oh no, it totally destroyed our definition of a gene. The gene must code for a protein. Here we have a sequence of DNA that codes for a function, but the function isn't mediated by a protein. So we actually refer to, you may see people talk about structural genes. Structural gene is of the 3 classes, RNA, tRNA, or mRNA. Messenger RNA is for structural genes. You make proteins off of them. So that's that. Yeah, so they said 1 gene, 1 protein was the original thing. People used to walk around and say, 1 gene, 1 protein. You have a gene, you make 1 protein off of it. They found transfer RNA, and they're like, oh, 1 gene, 1 function. Could be an RNA that does the function. I think we've gone over this stuff before. You probably went over this in general biology 2, or general biology 1, about once you find the ATG. I want to describe to you the process of how this all happens. And I want to explain to you the Kozak consensus sequence and why that was important to look in your messenger RNA. All right? Yeah, here's one of these things. There's a little asterisk there. One of the reasons that many people believe that all life on Earth is related and has arisen from an original cell that developed this translation code is all life on Earth uses the exact same code, whether it's bacteria, archaea, or eukarya. We all use CCU, all codes. Every bit of life on Earth will put proline into that codon, except a few unusual examples. Mitochondria use different things. A couple of the codons are different. Okay. The start codon defines the reading frame. So I'm going to erase this so I can draw 1 messenger RNA here real quick. Oh, I shouldn't have done that. Nuts. Of course that happened. Okay, no, we'll do it this way. 7-methylguanine cap. 7-methylguanine cap. You can say it enough, you'll get it. Poly A tail. AAU, AAA. Unless we do the CCU. GGG. CAU. Okay. So this is methionine. This codon is methionine. The next codon is, we said, proline. GGG. Oh. I gotta do a quick translation here. GGG. You guys know how to read this? First base is G. Second base is G. Third base is G. That's glycine. This is my termination codon, right? CAU. C, A, U is histidine. U, A, U. U, A, U is tyrosine. And then UGA is a termination codon. Right there. Okay. I put some additional code up here. I want you to notice something. Here's methionine. CUA. CUA is leucine. UGC. Cysteine. CUG. Leucine again, huh? GGC is glycine. AUU. Isoleucine. See how I'm using the same nucleotides and I'm getting a different code? So, depending upon which frame or which ATG is being used, AUG is being used, which start codon is being used, I'm going to get a different protein sequence. So, how does a ribosome know to use this one here? How does a ribosome know to use that and not this one? If this is the protein we want. And that has to do with the Kozak consensus sequence. So the way this all works, the ribosome consists of two parts that you should have learned in General Bio 1. There's the small ribosomal subunit and the large ribosomal subunit, right? This is about 20 proteins, this is about 40 proteins. There's like three ribosomal RNAs in here. I think there's one ribosomal RNA in here. I don't remember the specific details on it. There are other things that come and go. It's about 80 proteins totally involved in this. So you probably learned it as a machine that goes running down. And this is how translation is broken down into 3 steps, the same as transcription. There's translation initiation, there's translation elongation, and there's translation termination. In translation initiation, just like transcription initiation, there's a lot of things happening for that to proceed. And what originally happens is these are not together. The ribosomal RNA binds to a transfer RNA that has your initiation methionine. Remember we draw it in this structure right here? It's got that little cloverleaf structure. So this actually is the anticodon right here. The 3 nucleotides here match and are anti to AUG. They're the complement of that. Let's put our poly A binding protein down here. Protect our 5' end. So this is protected. This is protected because we have our 7-methylguanine cap. Turns out we don't trust that to be enough. We have a cap binding complex, about 4 to 6 proteins, I can't remember. These proteins are basically a promoter that attracts this to the messenger RNA. And so the small ribosomal subunit interacts with the cap binding complex and gives us the small ribosomal subunit with our messenger RNA, with our transfer RNA associated with it. The small ribosomal subunit then scans down the messenger RNA, trying to hybridize this to its match. And as it slides down, here it encounters an AUG. And depending on the sequence around that AUG, it can be recognized or not recognized. If the AUG is not in a Kozak consensus, for the purposes of this class, it slides right past. It can't hybridize to that. Only if the sequence is in a good Kozak consensus. And I guess I didn't do a good Kozak consensus. Change that here. We're going to make that G and we'll make that an A. Because I know that's the Kozak consensus, right? It changes what these code for, but anyway. So once it hits an AUG in a good context, in a good Kozak consensus, then it says this is the start. It begins translating from there. Does that make sense? Now you understand the importance of Marilyn Kozak's work in figuring out the Kozak consensus. Once this thing slides down, it's called scans down, and this hybridizes onto the initiation codon. The large ribosomal subunit then comes in, the small unit's here by now, and we begin the process of translation, elongation. Okay, any questions on this stuff so far? Am I going into unpleasant detail on this? Yes? No? You wouldn't believe the detail I went into grad school. It was sickening. People have studied, transition states and chemical reactions and stuff. It's pretty rough. So, I guess in this one I was talking about reading frame. I hope you guys got through that in General Bio 1. So we'll skip over that one in this class. You should know the difference in reading frames. I think I showed that up there. Okay. I'm not going to talk about how they, this is all General Bio review stuff. All right. Wow, it's in big text also. By convention, an amino acid sequence is always written N to C terminus. So, an amino acid, to remind you, is a nitrogen with 2 hydrogens over here, carbon, and an acid group here. And depending what groups are off here, that determines the amino acid. We call that the R group, right? So the next amino acid will be brought in. And the reaction will occur between this nitrogen and oxygen here. So there will always be an N on this end, and there will always be an acid on this end. So as we add our amino acids on, the singular letter code for this is methionine, proline, glycine, histidine, tyrosine is Y, and we have a termination codon. If someone writes a sequence out, you will always know that this is the N terminus and this is the C terminus. The same way that if I wrote out the nucleotides, you know that that's the 5' end and that's the 3' end. Okay. Proteins are built 5' or N terminus to C terminus. It's just convention. That makes sense. If you know 5' to 3', this makes sense. Again, proteins, once they're made, they fold up into secondary structures, like the alpha helix, the beta sheets, these turns and twists. How these all fold up to each other is the tertiary structure. Proteins then interact with each other. It's a quaternary structure. You should know that already. Yeah, Crick was actually a pretty bright fella. when they were trying to figure out, well, how is the code deciphered, Crick said there has to be something that can read that code. He predicted that there must be something, and it was eventually discovered that transfer RNA was that adapter, is what he called it. So we write the names out. This is transfer RNA that carries phenylalanine, transfer RNA that carries proline, transfer RNA that carries methionine. If it's going to be used as the initiation codon, they put a little I after it for initiation. Any questions on this? Do you guys go over this in General Bio 1? Not in this detail. Not in this detail. Okay. So here's the secondary structure of transfer RNA. The anticodon is in this stem loop structure here. So it's 3 stem loops. The amino acid is on the 3' end up here, I think. I think they drew it different than I did. So the amino acid would be poking out here. This is the tertiary structure of this right here. So this is this stem loop. This is this stem loop over here, I think, and this is this stem loop right here. And they know the 3-dimensional structure of this. There's an enzyme called tRNA synthetase. For each transfer RNA, there's a different gene called tRNA synthetase. So there's tRNA synthetase for phenylalanine. And that protein recognizes the exact perfect shape of that phenylalanine transfer RNA, and will put amino acids only on that guide. So here we go, large and small. Okay, here we get into it. I think, do I need to turn off the lights so you guys can see these images better? In the back, can you see these all right? Okay. So this is after initiation. The small subunit has bound to the cap, scanned down and found the initiation codon. The large subunit has then come in, and transfer RNAs are starting to add nucleotides on, or adding amino acids on. So what happens is the ribosome goes down, and it has 3 sites. It has a P site for the protein site, A site for the amino acid site, and an E, which stands for exit. So here's the transfer RNA that has the nascent peptide, the growing peptide, the new peptide that's growing, the new protein that's growing. And it holds that protein in the P, or the protein, or peptide site here. That leaves a binding pocket for another transfer RNA to come in. And to the best of my knowledge, this is random. The transfer RNA comes in and clicks in here, and if the anticodon can hybridize with these next 3 nucleotides in the code, then it will be held in place. If it comes in and it can't hybridize, it will go away. So basically there's a 1 in 20 chance that the correct one will come in. So these guys are constantly going in and out, in and out. Eventually the proper transfer RNA comes in, that the anticodon hybridizes, is complementary and hybridizes with these 3 nucleotides, that it stays, and the ribosome then shifts its shape slightly and breaks the covalent bond between this transfer RNA and the peptide and shifts that onto the amino acid that's in this site, in the A site. Now this big long peptide chain wiggling its way through here is not a good fit, and it actually causes the ribosome to shift down so that these 3 nucleotides slip into this spot, pulling that transfer RNA with it, and it bumps this guy out and into this spot, because these 3 have shifted to here. So this is now in the exit site, and it's ready to be released. And what entered originally, just kind of randomly coming in, and got held in there, the amino acid tRNA, is now shifted here and has this connected on. At which point there's 3 new nucleotides in here, we can repeat the process, and this is translation elongation. That's why the codon is 3 nucleotides. This distance is 3 nucleotides across. For the purposes of this class, we will say that the correct transfer RNA is always the one in here. Go to graduate school, and you can learn that there's actually an error rate where the incorrect amino acid sometimes is added in. It depends on the organism. So here's cycle of this coming through. So we have a transfer RNA come into the A site. It hybridizes on here. This is a single amino acid. This is the growing chain. This properly hybridizes. Oh, this is wrong. This is termination release. Is that this? This doesn't show this properly, does it? This gets, yeah, here it is. Here's the arrow. It shifts this over onto here. Puts that down. So this is illustrating termination of translation. This code, there's no transfer RNA that binds to that, because there's no amino acid that's added in termination. There's actually a protein that comes in and recognizes those codons, and then it severs the covalent bond here, but it doesn't make a covalent bond onto itself. And with this gone, this basically goes floating away and can now float up on its own. That's how translation terminates. The protein that comes in is called release factor. Oh, there it is, release factor. I don't want to do that. That's bacterial. Yeah, here I show arrows pointing to, I give it a description and point arrows to each of these for you guys when you review for exams. That just explains what I've already talked about here in class. Okay. Where are we? That's the cat binding complex. All right, this is the last thing I want to talk about today. It's pretty cool. I'm going to illustrate something to you. Your whole life, my whole life, people have illustrated this process out this way, right? So the ribosome binds here, the small subunit binds here, scans down to the proper initiation codon. It's got to be the right Kozak consensus sequence. The large subunit comes in, translation elongation begins, goes down, translation termination, and everybody teaches that the ribosome falls apart, goes floating away, and off we go. I read, when I was in graduate school, what happens is, in my area, if you go into graduate school, nobody earns a master's. You join a PhD program, and that's it. You go right through to PhD, you don't earn a master's. If someone has a PhD in molecular biology, it means they failed out of graduate school, and they're given a master's as a consolation prize. Okay? So what happens is you take 2 years of classes, and then you take what's called a qualifying exam. That can be a test of factual information from your courses. At my university, they said, go out and find something that nobody's ever done. It cannot be in your area of knowledge. It should be totally unrelated to your work. Find some problem that needs to be answered. Figure out how you'll answer it. Write that up like an NIH proposal grant. Turn that into your thesis committee. And then there will be a 2-hour oral defense of that, where you say why that's an important project, why the experiments you're proposing are the right experiments to answer the question. And if you pass that, you're allowed to go get a PhD. If you fail, we'll give you a master's. Good luck. So I went out and I was like, okay, I can't. For me, the definition was I couldn't do anything related to viruses because I was working in a virus lab. So I searched and searched and searched and searched. And I found a paper. Here are the proteins that are involved in the cap binding complex. I found a paper where somebody found that poly-A binding protein was shown to physically interact with the cap binding complex. And so what I proposed is messenger RNA is not some structure like this. Inside a cell, messenger RNA is this. Because the cap binding complex interacts with the poly-A tail. So I proposed that messenger RNAs are actually held in a circle inside a cell. And I propose the value of this is, if this room is 1,000 units of volume, and we have 1 ribosome in it, we have 1 ribosome per 1,000 units. However, if this part is flipped around like this, once that ribosome binds, and it rides all the way down, if it goes all the way to the end of the messenger RNA, it goes all the way back to here. If the ribosome then falls off here, if this is one volume of space, and I have one ribosome, what's the concentration of ribosome here compared to what we originally had out here? It's 1,000-fold higher concentration, isn't it? Get back to a basic equilibrium. So this is what I proposed, is ribosomes. It's very, very hard, if you're a messenger RNA, it's very, very hard to get a ribosome bound to you initially, because there's so few ribosomes in such a large volume. Once a ribosome binds and it circles all the way around, when it falls off, there's a very high effective concentration of ribosomes here. So that once a ribosome goes around, it will get stuck and continually go around that messenger RNA again and again and again and again. And I proposed this concept called ribosome recycling. And the guys on my committee had never heard of it. They said it's the most original qualifying exam proposal they'd ever heard in their life, and they loved the experiments that I proposed. I actually was at a conference, and there were people they had everything they needed to run the experiments. They didn't know about it. And at the conference, I mentioned it to these people, and I said, if you ran your system, you could just do this, and you could actually see if ribosome recycling exists. I explained the concept to them. Within 2 years, they published a paper talking about ribosome recycling. And it's now, in your genetics textbook, a picture of circularized RNAs, and it's made it into textbooks. How cool is that? That was fun. All right, so that's it for today. We're over time. What are we doing? Do we finish up translation next time? Are we scheduled for translation again next time? Does anyone know? Okay.